Welcome back friends. Welcome to another lecture from Just Biology. In this lecture we're going to talk about spectrophotometer. If you see this spectrophotometer, we could see many variations in the spectrophotometer. But each and every spectrophotometer had their distinct usage when compared to the normal one. In this lecture I'm going to discuss you with a basic spectrophotometer like UV and visible. So if you see this UV visible spectrophotometer, the basic one, actually this is the basic one, and this actually works based on the calorimetry principle. The calorimetry means the reagents or the agent which you will use for the identification of the certain molecule which is going to react with that reagent, and those two will react each other, and that will give you the color. That color is going to measured by the system. That system will give you a value. That value is actually your compound concentration or that molecule concentration. This is what usually does by the spectrophotometer. Apart from this property, we can use the spectrophotometer for the concentration of the cells, that is live cells, which is in the sample. So some instance we can use such a way. So apart from this, all these things, we should know the actual process takes place inside the spectrophotometer. We know that the light should be provided, so that's why the spectrophotometer, the photo means the light which is provided by the system, so that we have the light source. So as far as the light concerned, so we should know the spectral line of the light. So here I'm gonna discuss the spectral line of the light now, then we'll move on to the later different parts of the spectrophotometer and how the light passes through it. Now let's begin with the spectra. So we know that, so actually the spectrophotometer is a UV and visible. So UV visible spec we are going to discuss. Now the thing is, so we have to draw the spectral line. So actually this 200 nanometers, so that is UV range. So here we have the visible range, so that actually the 800 nanometer, actually nanometer is a visible. So visible, this is nanometer. Now actually what range which gives the UV and what range which gives the visible light? So that is what we should know. So here I'm gonna make divide here. So let's say this is 400 nanometer. So this is actually a partition. So I made a partition here over here. Now the range which starts from the 200 to the 400, so we can say that they are UV range, isn't it? So we can call this range as UV, so ultraviolet rays. So they start from the 200 nanometer, they ends with 400 nanometer. Then usually actually this from the 400 nanometer, so let's use another color for the understanding. So from the 400 to the 800 nanometer is a visible range. Actually all this blue, red, white and all the colors, so the colors which we can see through the naked eyes, we can refer from the 400 to 800 nanometer. Now this can be a visible range. So this can be a visible range. Then apart from this UV and the visible, we have different we have different rays also. So we have a different range also. So below 200, so you can see some kind of radiation or some kind of particle, because it's normally a radiation particle. So we have here X-ray and the gamma ray as radiation particle, so that has the less than 200 nanometer wavelength. So here 200 to 400 the UV and the 400 to 800 is actually visible. So beyond the 800 nanometer you can see a kind of radiation, so that would be referred as infrared. So that is IR radiation, so that is above 800. So that is above 800. We can call them that ray as infrared radiation. So this is actually a spectral line. So this is the actual spectra of the light. Now let's move on to the next part, that is a schematic representation of the spectrophotometer. How this process will be takes place inside the spectrophotometer. So as I mentioned earlier, so they have got a lamp or light source. They should attach with a light source. So it's usually light source would be a tungsten lamp in case of normal spectrophotometers, that is UV visible. So they have got a tungsten lamp for the light source. So this can be a light source, like light source. So this is light source. Let's say this is light source. Now, so light will emit. So this light source will emit an enormous amount of the lights, and that reaches some part of the spec that is called a monochromator. So the next thing is the monochromator. The monochromator actually a box-like thing, actually a box-like structure, so that contains prism. So you know the prism actually. So actually this prism, what it is? Prism inside the monochromator. So this is a prism. Actually the prism will be assigned inside the monochromator. So we have the light source here. The light source will emit enormous amount of the lights. Actually the monochromator, the placing this monochromator just right after the light source is, we have to sort out the lights. So we have here, we can see an enormous amount of lights I said. So here you can see many lights which has got different wavelength. So that is from 200 to like 800 I said. So we have to sort out all those lights. We need only a particular light which can pass us through your sample only. So other light should be prevented. That's why we use this monochromator with the prism. So here we have this monochromator. So what actually the monochromator does, just now I said. So they will absorb a more amount of lights. So this is the light ray, and so what actually this prism, so let's focus on the prism. The prism will absorb lights which is emitted from the light source, and this light source based on the wavelength, so based on the wavelength, so they will diffract that lights. So that is the idea. So here the prism will diffract the lights which is emitted from the light source. So you will get a different bands of light here. So here you have the light. So only from those cluster of the lights, only a particular lights will be allowed. So that is the idea. So here diffractor light, you can see many lights which has got a various wavelength, so that present inside the monochromator. From those, only a particular wavelength lights only passes through the next chamber. That is just right after the monochromator. You can see a small box like thing, we can call it as beam separator. So actually what is the function of the beam separator? The beam separator, they will allow only the light, particular wavelength light only. So they will receive only the one kind of light that should have a same wavelength. So they can allow only the lights which has got a same wavelength. So not, let's say for example, a light which is comes from this prism like 470, 280 and 390. Let's imagine these three. So they can allow all of three, but they can allow only the one light. So not all this wavelength light. You got the idea, isn't it? So here the function, actually the beam separator. The beam means a cluster of light. A beam means a bunch of light ray. So one kind of light ray can pass only through the beam separator. So the beam separator will align the lights. Actually, beam separator will align the lights inside the beam separator, and we have the chambers, like you can see a two chamber, actually two holder, so which you can see in the spec machine. So one is for control and the one is for actually the sample. Actually this is control and this is sample. So the light which is emitted from this beam separator, the light which is emitted from the beam separator will goes to control as well as the sample chamber. So they have the two fate. Actually the light which is coming from the beam separator will have to go both control chamber as well as the sample chamber. So that is the idea. So here we draw like this. The light which passes through this area and the here light which passes through this area also. So here this is the scenario. The light which pass through the control and light which pass through the sample. Actually this is the process. We have the two chamber, but the chamber should be connected with the beam separator and different things. Actually this beam separator will have this tube like structure which is tend to connected with the control and the sample chamber. So that is the idea. So here you can see this both control as well as the sample is been connected with a particular thing. We can call it as detector. Actually the detector will be connected, so both of the control as well as the sample chamber. So that is the idea here. So what is the purpose of placing a detector over here? So you can raise the question. So what is the function actually the detector does? The detector will detect usually the light which absorbed by the sample as well as the control. So that is the idea. The sample which has got some sort of molecule, so the molecule which absorb, probably they will absorb when they get excited. So that excited molecule will absorb some sort of energy or the light, light energy. So that is being recorded by the detector. So this detector is usually connected with the computer. So this is the idea. The computer will do all the works and the value and the unknown things and every unknown value and so all mathematical expression and all, then they will give you the result. So this is a normally spec do. But in case of this, actually this spectrophotometer is a UV visible it's not conventional, though it is not conventional, but they haven't connected with the computer. So that is the major disadvantage that I would say, because we have to take the reading and we have to calculate by the manual way, then we have to plot the graph and all, and this is actually the big task for us. But more or less this is quite fine for the identification of the molecule. So this is the convenient method for the identification of the unknown molecule, or it's not identification, just we should know the concentration. If you want to know the concentration, we can go for the spectrophotometer. So this is a schematic representation of the, actually this I'm supposed to write here. This is beam separator. So this is the schematic representation of the spectrophotometer. Then we'll move on to the principle, how this process takes place and at what basis the process is being takes place inside the small cuvette. That is the important one which we want to discuss. If you look at the principle of the spectrophotometer, so we can say, they are actually the combination. So the combination of concentration, absorption or transmission, intensity and all. We have many things. We have to do a little bit of physics here. So here we want to discuss the first thing, a law which is called which we can call Beer-Lambert's law. So that is the first one we want to discuss when we speak about the principle of the spectrophotometer. So Beer-Lambert's law. So this is what we want to discuss. Actually, what Beer-Lambert's law states. So what they says actually the Beer-Lambert's law states that a molecule which is present in your sample will get energized by the light. Then during that stage, they are in ground state. When they are in ground state, the energy level is zero. The energy is zero. When they are getting energized, they will move on to the next level. We can call it as excitation stage. So during that period, ground state and the excitation state, so in between they will absorb. So they would absorb some sort of light. So that light will be directly proportional to the concentration of the molecule. So that is the idea here. So actually, we should know the two terms here. The sample, so this S can be a sample, and this is the blank. So B that represent the blank. So here we're gonna make something related to the physics. Let's imagine the light which absorbed by sample. The intensity of the light we can write it as I. So this is the first one. So the I means the light intensity absorbed by the sample, we can written as I. The same way we have the blank also. So let's just remember. So we have the blank. So the blank which absorb the light. So that we can say that. So they are I0. So this is the two terms we should speak about actually. When this I and I0 is equal, so that is the blank S and the B. So here we have first scenario, the S is equal to the B. So that is sample is equal to the B. In some instance, we can say this I is equal to I0 is equal to I. So that means there is no compound at all. So the both are the same, same solution. But some case this I is not equal to the I0. So in that case, so we can be write it as, so here I divided by I0. So so we should subtract blank from the sample here. So that is the idea. So that's why I have written as I by I0, because we have to subtract this I0 from the I. So this I divided by I0, we can term this, we can convert this term into the transmittant form. So that is transmittance. The T means the light which is passes through the amount of light which is passes over the sample or the blank, or we can call it as transmittance actually. So what actually the T? The T is an amount of light which is passes through the sample as well as the blank. let's consider this equation. So I have told you, so they are actually this I. This I is the light absorbed. So this is absorption. So this is absorbed by the molecule which is present in the sample. But in case of I0, this is not absorbed actually. This is not absorbed. So what we can do here, we have to convert this formula. So in terms of absorption. So how do we convert this formula in terms of absorption? So before converting this formula, so why we need to convert this transmission? Actually this is percentage of transmission. So what is the demerits, or why we are not preferring this transmittance for identification of the unknown concentration? So that is the a question here. So why and what reason? What reason? So we are prefer this percentage T. So there is a reason. So the reason like this, let's consider this graph, and the x-axis we can plot absorption. So this can be absorption and this can be a percentage of T. So let's consider. But if you plot this graph by using absorption and the transmittance value, so let's consider transmittance value and the absorption value. So you will get a graph like this. So this is normally we can call this graph as declined. Why? Because so both are declined, but this is inverse graph. So if the percentage of T which is increasing, so thereby the decrease of T. If it is T is increasing, so you can see the A obviously, and the gradually A will be decreased. So this kind of graph cannot be used for the identification of the unknown concentration. So that's why, so we have to change. So we should make some rearrangement in terms of transmittance. How we can change this formula? Then that is the thing which we want to discuss now. So let's erase this part for so let's erase here. So we know that the percentage T is equal to I by I0. So this is what we know till now. Then what we can do, so we can do one thing here. So we just include, so we just convert this transmittance in terms of log. So in terms of log according to Beer-Lambert's law. So what would be the equation? So equation would be the T which is equal to 10 power minus concentration into the length. So what does it mean? So what does it mean? Actually the transmittance is equal to the 10 to the power of minus concentration into the minus concentration into length. So this is what actually the transmittance will give you. Then now what we can do, we have to convert this formula into the log form. So according to Beer-Lambert's law, then what would be the equation? So let's the absorption. So what we are gonna see, so we have to identify the absorption. So for the absorption we can rearrange this formula into the absorption form. So we have to include, or we have to convert this formula in terms of log. So log into, so because here using 10, so that's why we took log here. Then concentration into length. So this is what we'll get. So absorption is equal to log into concentration into the length. So here we derive the equation. So we have derived here. Then though we derived this equation is not sufficient for getting the information what we are looking for. Again we have to change, we have to rearrange further for the exact value. We need to get the exact value what we are looking for. For that we have to remove this log. Why? Because logarithmic calculation is more difficult when compared to the normal. That's why we have to remove this log, or we have to avoid this logarithmic form. For the removal of this log, so we have to take some constant term. So actually the constant term we have to take. So what term we should take here is, we should take epsilon. So epsilon is a constant, so which we can use instead of this log. That's how we change the equation. So what is actually this epsilon? Epsilon is a molar coefficient. So molar extension coefficient. So this is what? Epsilon. If you reframe this equation with this epsilon, you will get absorption, that is A is equal to epsilon into concentration into the length. So this is what we get. So epsilon is equal to, A is equal to epsilon into concentration into L. So this is what we get. Then if you see the equation now, this is not a desirable equation what we are looking for, because they are actually we framed like in terms of absorption. So we have to rearrange, actually the further we have to rearrange this equation. So how do we rearrange? Just what we need to calculate, we'll keep that alone in right hand side, and the remaining things will be we should take. So we should take to the left hand side. So that is what we have to do. So here we have the absorption, so just keep it as such. Then what we need to get it, so we need to know the concentration of the unknown sample. So what we need to know? So we need to know the unknown concentration of this sample. So just keep the concentration term in right hand side, or you just keep it as such. Then what is remaining in right hand side, just keep together and just divided by A divided by epsilon into L. So this is what usually do. So I'll tell you once again. So what we need to know? So we need to know the concentration, how much amount of the molecule is present inside the sample. So that is what we are looking for. So just keep the C, just keep the concentration here as such, that is right hand side. Then take length and the epsilon, that is constant here left hand side. Then you will get an equation like concentration is equal to absorption divided by epsilon into L. So this is what we get. So this is the equation for getting the unknown concentration from the samples. So this is actually a mathematical expression, the mathematical derivation of the Beer-Lambert's law. Then this is more than enough to understand actually what Beer-Lambert's law states. Then apart from this, what are all the application, what are all the significance, what are all the uses of this spectrophotometer that we want to discuss now. Actually this spectrophotometer is used for analyzing the molecule, that is analyte. So if you see any industries like medical industry, pharmacy, like research field, any field, if you take related to the the drug related fields or the analytical fields like which I mentioned earlier, so you can see the uses of spectrophotometer. But we have a variation, we have different classes of spectrophotometer is available now, but this is the basic one and this is most efficient one also to identify the first preliminary analysis. I hope this video is helpful to understand the concept. So that is spectrophotometry. If you like the video, please hit the like button and don't forget to subscribe my channel and share with your friends. Don't forget to click the bell icon for the latest videos from the Just Biology, and stay tuned and keep watching. Thank you for watching us.